Acute Lymphoblastic Leukemia. Let's get into it. So ALL is the most prevalent of all leukemias. I've decided to focus specifically on this type for this report due to its complex nature and the fact that it diverges from other leukemias in both form and function. So acute lymphoblastic leukemia is a blood cancer. Acute, in medical terms, means early onset, recent onset, or occurring in younger patients. Lymph relates to the stem lymphoid cells, white blood cells, that essentially have a malfunction that keeps them at the blastic stage. This means the cancer occurs in younger, more immature cells, and they lose their ability to differentiate into their respective roles as white blood cells, but they retain their ability to replicate. Risk factors for the mutation. So, there are many potential risks that cause the somatic mutation that cause ALL, though the precise, specific way the mutation occurs and why the mutation occurs is still unknown to science. Remember, it being somatic means that it can't be passed down. So, exposure to high levels of ionizing radiation, whether that be from medical treatment or something like Chernobyl, exposure to certain chemicals, including chemotherapy medicines, an infection with the human T cell lymphoma virus, which causes this mutation. Certain genetic syndromes, including Down syndrome, increase the risk of developing ALL. It's more common in males than it is in females. And over an individual's life, rates of DNA mutation grow. And this is the reason why older people, 60 plus, are more at risk of developing leukemia. Process of the mutation. So there are two primary chromosomal aberrations that develop to create the cellular effects characteristic of ALL, which is the inability to change into specialized cells but keep replicating as useless stem cells. So firstly, we have the translocation between the 12th and the 21st chromosomes. A weird fusion gene is created called the TELAML1 fusion gene. So this is most common in children and has a better prognosis. There have been a lot of studies into it to describe specific properties of the fusion gene, but a thorough understanding of its function is lacking. However, what we know of it is that it prevents this differentiation, but it retains the cell's ability to replicate. Scientists have been able to create a gene therapy that cures a very similar protein that creates the EML1 cell line, allowing these cells to continue and differentiate correctly. The only problem is that the AML1 cell line still shows impaired differentiation capacity, and a cure to this has not been found. Next, we have the 922 translocation. So this is between the 9th and the 22nd chromosomes. So this is known as the Philadelphia chromosome. The BCR-ABL fusion gene is created as observed in the diagram. So this has a far worse prognosis and is more common in older people. So within leukemia cells, various sporadic mutations can occur as they reproduce at such a rapid rate, including point mutations like deletions or expanding trinucleotide repeats. These mutations contribute to the dysregulation of cell growth and differentiation, promoting the development of a more aggressive leukemia. So, in conclusion, we have all these cancerous, compromised white blood cells or lymphoblasts descended with this somatic mutation being pumped out by the machinery within the bone marrow, and they proliferate into organs and the peripheral bloodstream as they cause leukostasis, which is the negative side effects of leukemia. They metastasize throughout the body and they cause death. Remember, because the mutations that cause ALL are somatic, specific genes that will promote the cancer are not passed down to offspring. However, certain risk factors may potentially be passed down. Anyway, thank you very much for listening. I hope you learned a little bit more about ALL and stay tuned. Cheers.